Good morning to Dr. Aina and my fellow classmates. We are from Group 5 and we will be presenting on immunosurveillance. So, before I brief about immunosurveillance and immunosurveillance effector, I will give a situation to give a better understanding on what we are going to talk about today. So, to protect our country, we need a system, or else anyone can enter and do anything they want, right? So, we have a national defense system to protect our country. But the system alone is not enough. We need people to work under it to protect our country. So, for our body, the same system applies here. In our body, immunosurveillance is the national defense system, and immunosurveillance effector are considered as the police and army men who hunt for the unwanted people who is entering our body. So, by theory, immunosurveillance is an immune system that patrols our body to recognize and destroy any foreign pathogen and cancerous host cell which will cause harm to our body. Effector cells are considered as relatively short-lived activated cells that defend the body during an immune response. Effector B and activated T cells, including cytotoxic T cells and helper T cells, are an example of effector cells. Now, moving on to types of immunosurveillance effectors, the first type is natural killer cells. Natural killer cells are considered as the primary defense line. They are responsible for killing tumor cells and they are assisted by interferon IL-2. The differentiation and expansion of NK cells from CD34 plus hematopoietic stem cells are regulated by early activating cytokines which interact with respective receptors, and they are responsible for hematopoietic stem cell proliferation and differentiation. This process is very crucial for NK cell survival, anti-tumor function, relative signals, strengthen activating or blocking inhibitory cells. These are vital for improving NK cell efficacy in adaptive cell therapy as well. Now I'll pass to the next presenter.
Next, I will explain about another immunosurveillance effector, which is B cell, and how it responds to infection. In response to activation signals, naive mature B cells will proliferate and differentiate into effector cells. Induced regulatory B cells will secrete interleukin 10, 1735, and transforming growth factor beta. Short-lived plasma cells that secrete polyreactive antibody can also be generated. Sustained B cell activation leads to further differentiation and selection in organized lymphoid structure called germinal centers. This will lead to activation of nuclear factor kappa B and upregulation of activation-induced cytidine deaminase to induce affinity maturation of antibodies and differentiation of specific long-life plasma cell and memory B cells. Next immunosurveillance effector is helper T cell. Helper T cells are activated by the antigen-presenting cells and differentiated into different subtypes, which are helper T1, T2, and T17 cells. The subtype helper T cell will activate and other cells to generate particular immune responses. In helper T1 cells, it plays an important role in immunity against intracellular microbes and involved in type 4 hypersensitivity reactions. In helper T2 cells, it is important for immunity against parasite, predominant in allergy, and involved in type 1 hypersensitivity. In helper T17 cells, it plays an important role in the defense against extracellular pathogens and involved with autoimmune and inflammatory disorder. I will pass my turn to Firdaus.
Right, thank you, Julian. So I will continue with how immunosurveillance will against cancer cells. So basically, I will focus on cytotoxic T cells. All right, first of all, I need you guys to know about what cells that involve in this process. So the first one is TC cells, which you can see in the picture here. On the TC cells, we have the TC receptor, which called TCR. Right. On the other hand, on the cancer cell, we have MHC class 1 molecules on the cell of the cancer cell, and also the antigen. All right. when the TCR bind to this antigen, boom, they will induce the first signal whereby it will activate the cytotoxic process of the T cells. So now I will explain about how the cytotoxic process will occur. So the first one, the TC cells, which is the cytotoxic cells, which is the police in this case, will find the target cells, which is the cancer cells. All right. So the interaction of the TCR, which is the TC receptor on the TC cells, will interact with MHC class 1 and antigens on the tumor cells. They will secrete PNG. What is PNG? So P is perforin, where it create a pore, and G is granzyme, whereby the granzyme is a toxic enzyme that will induce apoptosis. So there will be also the second signal arise, whereby interaction between APC, antigen presenting cell, and also the CD28 on the T cells. So this action will produce cytokines called interleukin 2 or IL-2. So as a result, this second signal, we call other T cell to help and also initiate T cell proliferation, differentiation, and also survival. And also I will explain about the main difference between the cytotoxic T cells and also NK cells. So basically the main difference is the activation, whereby the NK cells no need MHC class 1 to activate the process, whereby the TC cells need MHC class 1 to induce the process. Right now, I will share to you guys a video, right, on how TC cells will kill the cancer cells. Right, so let's watch the video.
These are immune cells called cytotoxic T cells. Cytotoxic T cells are able to recognize and kill virally infected and cancerous cells in the body. These T cells are on patrol, searching for their targets. Let's take a closer look at how they move. Cytotoxic T cells are constantly migrating around your body.
Yeah, so this is the
They move through your tissues by pushing out the membrane at the leading edge of the cell and pulling themselves forward. When a T cell encounters a cancer cell, an explosion of membrane protrusions explode the surface of the target.
Two things, P and G, which is perforin and granzyme.
Cytotoxic T cells kill their targets with toxic chemicals. The chemicals are housed in these red structures called lytic granules. You can see the lytic granules polarized from the rear of the T cell to the blue cancer cell. These toxic granules secrete specifically toward the target, protecting innocent bystanders which may be nearby. As the membrane of the target is compromised by the toxins in the granules, our indicator flashes bright red, signaling the impending death of the cancerous cell.
Alright, so basically that's all from our group. Thank you.